Amino acids play a crucial role in helping the cell live in many organisms. They are considered the building blocks of proteins, and once the proteins are formed, they have many functions, including being an enzyme, being a part of the cell membrane to give it structure, helping the cell undergo metabolism, and just many other processes that help the cell live. The structure of the amino acids has 4 main groups. One of them is amino functional group, which is basically just an NH2 group. The second one is a carboxyl functional group, which is a COOH group. The third one is a hydrogen, and the fourth one is an R group that we'll get into in a little bit. The first 3 points on this sheet are common to 20 amino acids that we'll talk about. The R group is the variable group and really helps determine the type of reactions that the amino acid will undergo. Now, these classifications can vary from book to book, but these are the 4 ways that I learned them, so I will explain the categories accordingly. So one of the categories is acidic, the second one is basic, then you can have polar or hydrophilic amino acids, and then lastly you have nonpolar or hydrophobic. Hydrophilic amino acids are hydrogen loving. The word philic means loving, so you'll mostly find them in areas where there's a lot of hydrogen or water available. Hydrophobic, on the other hand, are hydrogen fearing, so you'll find them mostly in areas where there isn't a lot of water available, so the interior membranes of the cell membrane. And then the R group basically determines the type of reactions that an amino acid will undergo. The structure of an amino acid is very similar to this picture right here. You have your carboxyl group, you have your R group, you have your amino group, and then you have a hydrogen. This carbon right here is called the alpha carbon because it's right next to the carbonyl of this carbon, and it is chiral because there are 4 different groups attached to it, and those 4 groups would be the amino group, the R group, the hydrogen, and the carboxylic acid. There are many amino acids present, but only 20 of them can be coded by the human genetic code, and those 20 amino acids are called proteinogenic amino acids. Now to the stereochemistry of amino acids. Amino acids can either be classified as L, S, or D and R. If the amino group is on the left, it's called an L amino acid. If it's on the right, it's called a D amino acid. D stands for dextro, which is right, and then L stands for levo, which is left. In order to determine if something is L, S, or D and R, you have to determine a chiral center. A chiral center is basically a carbon attached to 4 different groups, and those groups can be anything as long as they are not the same thing. They could be a hydrogen, a carboxylic acid, amino, and an R group, which in our case is chiral. Almost all amino acids are S configuration when you assign priority, except cysteine, because in that case your sulfur group would take priority over your oxygen. And glycine is one exception in that it does not have a chiral center. Special amino acids include glycine, proline, cysteine, and histidine. Glycine, which I will show the structure to in a couple of moments, does not have a chiral center. Proline bonds with itself, so you would not want it in your alpha helix and protein or beta sheets because it introduces kinks into the structure or your secondary structures. Cysteine is unique in that it is an R amino acid instead of an S because of the S that takes priority over the oxygen group. And then you have your histidine, which is positively charged at pH 7, that I will get more into when we discuss about pHs and pKa's and PI's. So this is the structure of the amino acids. I have the R groups in purple. So glycine, the R group, is an H. So since this carbon right here, the alpha carbon, is attached for two of the same groups, it cannot be considered chiral. Proline, on the other hand, kind of bonds with itself, forming kinks into secondary structures. Cysteine is right here, and due to this SH group, you'll notice that it can form really strong bonds in the tertiary structure called as disulfide bonds. Histidine is unique in that at physiological pH, its pK is really close to 7 or 7.4. So one of these nitrogens would be protonated, giving it a plus charge.